All right, in this video, I want to spend some time talking about what's really happening when we're breaking down some major nutrient groups as we go through the digestive tract. So we're going to focus on how we break down proteins, carbohydrates, and lipids. So starting with looking at where our major digestion is going to happen, or at least where we secrete things into. So we're going to focus for each of those three things on three locations. So we're going to take some time to look at what's happening in the oral cavity, what's happening in the stomach, and what's happening at the small intestine. And with the small intestine, we're actually going to split this out into two parts. There's also going to be what's happening in the lumen. So what are you secreting into that space? And then also we have this part that's called the brush border. And so the brush border enzymes, so we have this thing called the brush border. The brush border enzymes are going to be the enzymes that are built into the wall so we can do final breakdown before we absorb things. The nice thing about these is the brush border is holding the enzymes so you're not losing them out the digestive tract. You don't have to constantly keep regenerating them. All right, so let's first start our journey with carbohydrates. So with our carbs, starting in the oral cavity, we're going to have two of the three salivary glands that secrete an enzyme to break down carbohydrates, or complex carbohydrates specifically. So looking at breaking down starches. So the parotid salivary gland and the submandibular. So the parotid and submandibular glands are going to secrete salivary amylase. When we get to the stomach, that salivary amylase that we were using up in the mouth is going to get denatured because the environment inside of that stomach is very acidic. So we're going to have hydrochloric acid in there that's going to denature or turn off the salivary amylase. So when we get to the small intestine, we got to pick it back up. So at the small intestine, the pancreas will secrete, and specifically the acinar cells, will secrete their amylase, and so we'll call it pancreatic amylase. Now, in order to absorb, we have to get down to the smallest unit, and for carbohydrates, those are going to be monosaccharides. So to get to the monosaccharides, you have to break down disaccharides, two sugars. And so what we're going to have at the brush border is that breakdown of those disaccharides. So specifically, we're going to break down lactose with an enzyme called lactase. So when you're ending in ase, it's telling you it's an enzyme. When you end in ose, it's telling you it's a sugar. So we'll have lactase, maltase, which breaks down maltose, and sucrase, which breaks down sucrose. And that'll get us all down to our monosaccharides, which then we can absorb. So that's what's happening with our carbohydrates. Okay, so next up, let's talk about what happens with our proteins. Inside the oral cavity, we'll have some mechanical processing. We're chewing, but we don't secrete any enzymes for breaking down proteins all the way up in the oral cavity. But at the stomach, we're going to have the chief cells secrete into the space an inactive protein digester. You can't secrete it in the active form because remember, most of our stuff is made out of protein, so you would start digesting yourself. So we'll secrete an inactive enzyme called pepsinogen. When you see gin at the end, it's telling you it's inactive. So to keep track of our inactive stuff, I'm going to put a bubble around it. So it's in a cloud. It's being thought of, but it's not all the way there, I guess is kind of how I'm thinking of like a cloud. And so we're going to turn it into pepsin, the active form, by the hydrochloric acid. That hydrochloric acid was secreted by the parietal cells. So parietal cells and chief cells, they're part of the gastric gland cells. So they're in the wall of the stomach secreting into that lumen. The parietal cells are going to make the environment acidic. The chief cells make the enzyme, so they're going to secrete the inactive form of the protein digester. It's going to hit the acid, and it's going to change shape. Because most of the time when we have an acidic environment, when a protein hits it, protein changes shape, and it denatures it into a form that doesn't work. That's what happened with the salivary amylase. Here, we're secreting it in a shape that doesn't work already, and so when it denatures, when it changes its shape, it changes it into a form that does work. So now we can start breaking down those proteins. Same thing at the intestines here. We're going to secrete some digesters for proteins, but we can't secrete them in their active form. So these will be our proteases. And proteases are, again, inactive. They're going to come from the pancreas, acinar cells specifically. So let's add that in here. And so they're going to secrete these proteases. These are going to be things like trypsinogen. The gen's going to tell you it's inactive. Chymotrypsinogen. And another way you can tell something is inactive is we can use your pro. So pro-carboxypeptidase. And so all of these are inactive. All right, now how do we activate them? This is going to be something different. So we don't have the acidic environment anymore. Once we get to the small intestine, we're going to have buffers. We're going to make the environment more neutral. So to get this to activate, it's actually a brush border enzyme called enteropeptidase, also called enterokinase. And so that enteropeptidase is actually going to activate our trypsinogen, and it will turn into trypsin. So that's the enteropeptidase. It's built into the wall. So as you run past that, sitting on the wall, that's where you get the activation of trypsin. Once you have trypsin, trypsin's going to activate these two. So trypsin will activate the chymotrypsinogen, turning it into chymotrypsin. And then the procarboctipeptidase will turn into carboctipeptidase. Basically, you just take off the gin and you take off the pro part to activate them. Once we start getting down to small peptides, you got to break the peptide bonds. And so we're going to use peptidases for that. And those are brush boards. Again, you're going down to the final version. They're going to break it down into amino acids. Our last category, we're going to look at lipids. Perfect world, I would have left myself space for that. So we'll put it over here and pretend it's going to go over there. So with our lipids, in the oral cavity, you are secreting lingual lipase from the tongue. But it is inactive. So again, we're going to bubble that one. So lingual lipase is inactive. We're going to take it to the stomach. When it gets into the stomach, it hits that hydrochloric acid, and that's when it gets activated. You're also going to secrete into the stomach a gastric lipase. And that is coming from those chief cells again. So chief cells over here, we're secreting the inactive form of the protein digester. They also secrete the gastric lipase so that they can break down lipids at the stomach. When we get to the small intestine, pancreas once again is a big player. And so the pancreatic cells are going to secrete a pancreatic lipase. But also to help with protein digestion or lipid digestion is that you need something to separate them, because remember, fats or lipids don't like water. So as you're going along the digestive tract, these fats are clumping together and they're going to make a big circle around each other. And you can't have enzymes work on them if they can't reach them. So what you need to do is you have to have a way to separate these fat droplets into smaller droplets. So if you kind of picture, if you put water and an oil in a water bottle and you shake it up and then you can see all these small little droplets separate, that's what's going to happen through a process called emulsification. We do that using bile. Bile comes from the liver. That's where it's made. But the gallbladder can also secrete it. So if you're not using it, you'll go store it in the gallbladder if you have one. So we have the liver and gallbladder secreting bile, which is an emulsifier. It will separate the droplets. So it's not actually breaking the bonds. It's not actually breaking down the lipid. But what it is doing is it's allowing you to have increased surface area. So all this lipase that you secreted can do its job. Now, there isn't any brush border enzyme for the lipids, because lipids are actually able to go through that cell membrane if you get them to a small enough size, because they are lipid friendly, and phospholipid bilayer gets along with lipid friendly things. So this is kind of an overview of how you're breaking down some major things, starting with carbs, releasing salivary amylase in the oral cavity, pancreatic amylase in the small intestine, and then some brush border breakdown. Proteins, nothing in the oral cavity, secrete into the stomach and to the small intestine. The inactive form, activate them in the stomach with an acid, and in the intestines with a brush border enzyme, and then a final breakdown with a brush border enzyme to get to amino acids. Lipids, going to release lingual lipase. It doesn't work until you get to the stomach. That's also when you'll add gastric lipase. Get to the pancreas, add pancreatic lipase. But that addition of bile to emulsify and separate out the fats is really where you can start picking up some speed and breaking these things down. All right, hopefully that was helpful.